Hello, everybody. Welcome back to Creation Myths. I got a really exciting one tonight, I think. As you can see, my guest tonight is Will Duffy of the Final Experiment fame. Before I say anything else, I just want to make sure that I can see and hear myself okay on YouTube. So let me just make sure that my window looks okay and everything looks okay on my end. It does look okay, and folks, you may notice I am a person tonight, not a Minecraft character. I've been having resolution glitchiness, but it looks okay so far tonight, so hopefully it stays that way for the duration of our conversation. So now that the technical stuff is out of the way, chat, if there's any issues, let me know. But what we're doing tonight is kind of a spin-off. If you've been following the ongoing conversations Will has been having with Erica Gutsa-Gibbon, doing kind of an intro to evolution as a field, this conversation grew out of their last one where junk DNA was mentioned very briefly, and Erica, like in passing, was like, by the way, Dan's in the chat, talk to him if you want to talk about junk DNA. So I reached out to Will, and here we are to talk about junk DNA. So, Will, let me give you a minute to introduce yourself, and then we'll get going. Take it away.
Yeah, thanks so much for having me. I created the Final Experiment, which took flat earthers to Antarctica a year ago this month, last December, to show them that we indeed live on a globe. And that ended up leading me to a mutual friend introduced me to Erica Gutsa-Gibbon. And so I had a bit of a humility moment through the Final Experiment, which was telling flat earthers they're critical of the globe, despite knowing almost nothing about the globe, realizing I'm critical of evolution, even though I don't really understand evolution. So I'm getting an incredible education on evolution from Erica. I think she's definitely one of the best, if not the best, on YouTube. And then our last discussion, this topic of junk DNA was brought up, which I know very little about, but I've heard about it for years. And so Erica then introduced me to you, Dan, and here we are. And I'm looking forward to talking about junk DNA, trying to understand your position. I'll have a lot of questions, and let's go from there.
Let's do it. So, before we get into it, folks, let me just say I'm going to put up some slides here. So let me add those. Just a little short, I want to emphasize short slideshow for us tonight. For anyone expecting this to be another like 4-hour extravaganza, that's not what we're going to be doing. I promise we're not going to do another 4-hour thing. This is a short little spin-off, this little bonus episode. It's not going to be a full big thing. That's Erica's gig here. So what we're going to do tonight is we are just going to briefly introduce the concept of junk DNA. We're going to look at this in the context of the human genome specifically. So we're going to start with part one. What is the human genome? Just like, what is it made of? And then we'll zoom in and say, okay, that's what it physically is. So what's actually in it? What are the different regions? What are the different parts? Then we'll look at those parts and say, okay, of all of those different things we can annotate the genome with, what are they and what do they do? So we'll say how much of that is functional, how much of it is actually doing something that affirmatively contributes to the physiology of the cell or the organism. And in going through that section, we'll look at the regions that we know are functional, that we have direct evidence for, the regions that we have maybe circumstantial evidence for, but it could go either way, and the regions where we can be really sure they're not doing any of the functions that we've identified in the genome that it does. And then part four, we'll kind of put all this together, why most of the genome is junk. We'll kind of look at this from a couple different directions. And we'll wrap it up with a short summary, which is part five. And that'll be a single slide. So this will be not super long, not super detailed. This is meant to be kind of an introduction. This is not a detailed kind of thing. If anybody wants the detailed case, I have two recommendations for you. First, I've got a book by Dr. Larry Moran, who does the Sandwalk blog. It's called What's in Your Genome? 90% of Your Genome is Junk. And if you want a book length, version of what we're going to talk about here. This is it. It goes through every aspect of your genome in painstaking detail. It's very clear, very concise writing. There's just a lot to talk about. And that's a really good resource. And then something that I need to link down below that I'll do after the show is Dr. Zach Hancock, whose channel is Talk Pop Gen, has a very detailed video on this topic where he goes through it. And he also has an interview with Dr. Moran. So those are all good resources that I can recommend on this topic. So all that stuff out of the way, let's get into this. Let's talk about the genome and junk DNA.
Hey, Dan, real quick.
Yeah, Sorry. Go ahead.
Someone in the chat named FSM Dog just said, if you take it away, nothing happens. Is that true?
So there have been experiments in mice in particular where they've looked at regions where they're saying, is this region functional or not? Could go either way. So they just did like the deletions of millions of base pairs of DNA, and turns out it was fine. Now, those experiments are limited in terms of what they are because we're not deleting 30% of the genome at a time. They're deleting, I think it's like 1% is what it's millions of base pairs. So it's big chunks. I think it was like 30 million for one of the versions of it. Here, let me put this comment up on screen. Actually, let me. Here we go. Take away.
Yeah, it came up when you were talking about a book about our genome. So
yeah, so we haven't done this for humans because you can't, experiment on humans like that. But we have done this in other things, and the big example is mice, where we've deleted tens of millions of base pairs at a time and shown that there's no cost that we can document to that in those experiments.
Do you know how long they were? Like, I don't know what the lifespan is of a mouse.
Mice are pretty quick. You could do several generations in a year. And for those experiments specifically, I don't know off the top of my head how many generations they did, but they showed that the offspring of the deletion mice were fine and could have kids of their own. So I guess grandkids. So they carried it that far at least. I don't know if they went past that.
Okay. Great. Thank you.
So we'll start with what is the genome? In other words, what's it made of? So we'll start with just like DNA. Deoxyribonucleic acid is the molecule. And at the bottom of the screen, we have just kind of three ways that DNA is represented. You've got kind of your cartoony twisted ladder on the left here. On the right, you have your more kind of like molecular space fill kind of model there. And then in the middle is zoomed in to looking at the actual base pairing that occurs across the rungs of the ladder, as they're often described. So you have your backbone, that's a sugar with phosphate groups connecting it, and then the inside of that twisted ladder is the nitrogenous bases. It's A, C, G, and T, adenine, cytosine, guanine, and thymine. You can see here, A pairs with T, C pairs with G, and, not to get ahead of ourselves, but it's the sequence of the letters that actually is responsible for whatever it is that DNA is doing. So the backbone is biochemically constant, and it's the sequence of those letters that dictates the stuff that goes on within the DNA molecule. So that's the kind of first level there. It's just made of DNA. Now, the genome is not all one giant piece of DNA. It's arranged into segments. We call those segments chromosomes. Here we have a human karyotype. This is just a visual of chromosomes here. So we have 46 chromosomes in 23 pairs. So you get 23 from mom, 23 from dad. Because we have two complete sets, which is to say two of each chromosome. So 1, 2, 1, 2, 1, 2, all the way down. This is called a diploid state. Just means you have two of each thing in your genome. So you have two of everything, means you're diploid. One complete set from mom, one complete set from dad. Now, in terms of the size of the human genome, a diploid human genome is going to be about 6 billion base pairs, a base pair being the rung in the ladder. So you got about 6 billion of those in a diploid set. A haploid set, which is just one set of everything. So the 23 chromosomes you get from mom or the 23 you get from dad are a haploid set on their own. And that's about 3 billion base pairs. So what we're talking about here in the human genome, as we go through like what the percentages are, how much each thing takes up, we're going to be generally talking about just like in a haploid genome, what are the percentages that we deal with? Obviously, a diploid genome is double the size, but the percentages are the same because it's everything doubled. But generally, when this stuff is talked about, it's talked about in the context of a haploid genome. It doesn't really matter for the numbers, but just in terms of the convention, that's generally what we're talking about. Okay, so that's what the genome is. So now all the different parts of a genome. And before anybody either chat or later on watches this and wants to say, come on, Dan, those aren't all the parts of a genome. I know. I'm going to leave stuff out. Like I said, the idea here tonight is a basic intro. We want to hit the high marks. We're not going to do, the book length thing. But we're going to hit the important parts of what's actually in the genome here. So first we have genes. Obviously, those are the parts that get all the headlines, right, is the genes. Those are the regions that code for proteins. So you have a region where you do this process called transcription. So it's DNA, and you make a complementary strand of RNA. That's called mRNA or messenger RNA. And then that messenger RNA goes through a process called translation. The ribosomes within the cells do this, and the translation results in a polypeptide, which gets folded into a functional protein. So we go from DNA to RNA to a protein, and that's the basic process of gene expression. When I do this in class for real, this one line right here, that takes 80 minutes, but we're not worried about the biochemistry. We don't care about the mechanisms. It's just that is the thing that happens with genes, and we can leave it with that for tonight. Now, genes are actually made up of two regions in the genome. You have the protein coding parts, which are called exons. Those are the parts that actually have the little bits that tell you the amino acid sequence for the protein. And then in between those are much larger regions called introns that are non-protein coding. So they don't tell you the sequence to make a protein. And in between transcription and translation, you actually cut those out. There's an RNA processing step in there where you get rid of the introns is one of the things that happens there. Several other things happen, but it doesn't matter for what we're doing tonight. But in terms of the genome, we've got regions that are genes that contain the information to make proteins, and also a bunch of stuff that doesn't contain that information that we cut that out along the way. Okay so far?
Yes. Yep.
Familiar? Anything from like 7th grade science class kind of ring a bell? This is...
Yeah, it's definitely coming back. You'll probably get to this, but I think one of the biggest areas of confusion was the assumption, and I think it was the assumption by everyone, was that the protein coding regions were the only thing that was really important.
Yes, so this is a common thing that you hear, right? It's biologists thought everything that wasn't protein coding or, directly associated with it, like regions called promoters that we'll get to in a minute, anything that wasn't directly involved with gene expression to make proteins was non-functional junk DNA. So that is a common thing that you hear. That is not true because the term, so short little history lesson, and if for anyone interested in the longer history lesson, Zach Hancock does the long version of this. But the term junk DNA first appears in the literature sometime in the 1970s. I forget the exact year, but it appears in the 1970s, describing DNA that doesn't have a specific function in the cell. But prior to that, in the 1960s and the 1950s, we had actually already directly documented regions of the genome that were both non-protein coding and clearly functional. So these are going to be things like promoters and other like gene regulatory regions, that basically the switches that control if a gene is on or off. And also what are called ribosomal genes, so things for making like, I'll talk about it in a few minutes, but tRNA, rRNA, these are other things that are important for the cell, but they're not proteins. So these are regions of the genome that are clearly functional, and we knew they were functional, and those were discovered and characterized prior to the term junk DNA actually being defined in the literature. So it has never been the case that we thought all of the non-protein coding stuff was junk. It's just the way the history lined up is the term appeared after discovering stuff outside of the protein coding that was functional. But you do hear that frequently, all the time. Yeah. But that is historically not correct. So the next thing we'll talk about, relevant, are the regulatory regions. So promoters are where you start gene expression, so it's where you start the process of transcription. So this figure is from the textbook I use for my classes. The figures are very minimalistic, which I like. So we've got a gene right here, and the promoter immediately adjacent to it is just the region where the enzymes that are going to do the process called transcription, they're going to attach to the DNA at the promoter, and the promoter sequence is very specific, and it says, like, start here with a giant arrow pointing next to it. Promoters, if you're thinking like on the highway, you know, they have like, it'll be like, McDonald's with an arrow pointing down. Like, that's what the promoter is. You don't do transcription of the promoter, but it tells you where to start, and it's immediately adjacent to it. So the enzyme grabs onto the promoter and then starts the process adjacent to it. So that's a regulatory region. And then you have regions called enhancers and silencers that are regions that modify the activity of the promoter itself. So what's showing on here is you have all of these like blobs represent proteins. And they're binding to the DNA up here. You've got these regions. In this case, it's enhancers. When you have proteins binding to enhancers and they interact with a promoter, that increases the rate of gene expression for that particular gene. You could also have regions called silencers, where when the transcription factors are the kind of catch-all term for those proteins, when the transcription factors bind to the silencers and interact with the promoter, they shut it down. So these are regulatory regions that are going to increase or decrease the rate of expression for the genes that they modify, right? And then this last thing here, methylation sites, has more to do with chromosome structure than regulating any specific gene, but it's still a form of regulation. So this requires a little bit of chemistry here, but methyl groups are just carbon and hydrogen, so they are hydrophobic. Basically, they're nonpolar. They don't interact with other stuff. They kind of just want to do their own thing. So they're like oil or soap, right? So think about oil and water. If you have like cooking oil in a pan, it forms those bubbles, right? Because it doesn't interact with the water. It doesn't mix with the water. It forms its own little self-contained things. Methyl groups want to behave that way, and the inside of a cell is basically salt water. So when you methylate the DNA, there are specific sites where you can attach these methyl groups. When you methylate the DNA, it causes the DNA to condense, right? It causes the methyl groups to associate with each other to get away from the surrounding water molecules. When you do that, that makes it harder to express the genes because the proteins physically can't fit in there. If the DNA is too tightly packaged, then the proteins can't, they got to elbow their way in, and they just can't. There's not enough room, so those genes don't get expressed. So the methylation sites, they're less specific than like an enhancer or a promoter, because those things affect single genes, whereas methylation is more of like a regional thing. In a chromosome, you can methylate a region of a chromosome and kind of shut that region down. And there's other similar things you could do. You could remove the methyl groups, you could do acetyl groups. It all kind of falls in the same category of regulating the structure of the chromosome. And in doing so, you're affecting the degree to which that region is available to do other things, right? So that's what's going on with methylation sites. So those are a specific sequence, and that's going to be regulatory in terms of affecting the things around it. So that's they're not long, but they're important for how the genome works.
Okay, very complicated.
Yeah, this is a lot, and this is the short version. So then we can look at the actual structural components of the DNA. So most people are probably familiar with representing a chromosome as like an X-shaped structure where you've got these two halves called sister chromatids that are just copies of each other. And they're connected at a region in the middle called the centromere. That's the region in blue, just where that X is kind of pinched in. And then at each end, you have the telomeres. It's kind of a protective cap at the end so that if the DNA degrades from the ends, which happens over time for various reasons, you eat away at the protective cap rather than like any actual genes that, you need the genes, but the protective cap is just a region that's there to kind of be the target before anything below it gets damaged just by the ongoing processes of being in a cell, being exposed to the stuff going on in the cell, and you lose a little bit each round of DNA replication. So we've got our telomeres and our centromeres as structural regions, and those have particular repetitive sequences that we can identify. And then you have what are called spacer regions, where throughout the chromosome there are some regions where the actual sequence of the letters doesn't matter, but the distance matters. So for example, actually I'm going to go back one slide to illustrate this. If you look right here, you've got your promoter right here, and you have your enhancers right there. You'll see how the DNA kind of bends around. We call that a hairpin bend in the DNA. That distance has to be just right. So like, for example, it could be 5,000 nucleotides between the promoter and the enhancers, and it's good. But if it's 4,000 or if it's 6,000, it's too short, it's too long, it doesn't line up when it bends around, so it doesn't work. So the sequence of that region doesn't matter, but you need the space there in order for everything else to work right. So it's the distance rather than the sequence. So those are, we'll talk about it, but those are obviously functionally very important. Even if the sequence itself is not responsible for the function, it facilitates the interactions of other components in the correct way. And then the last structural thing we'll talk about here has to do with proteins called histones. So histones are proteins that associate with the DNA, and the DNA wraps around the histones, and it's how we fit all the DNA in our cells. Because if you were to take all the DNA in any one of your cells and just lay it out end to end, it would be like 6 feet long. So obviously having this microscopic thread that's 6 feet long in a microscopic nucleus within a cell, it has to be very efficiently packaged in order to get it in there. It's the way I always think about this is every time I'm loading my car to like go to the pool with the family or go on vacation, everything is always nice and neatly packed in the bags on the way. And then it's really hard to fit it all in the trunk on the way back, right? Because everything is all over the place and it's not neatly packed anymore. And DNA packaging in the nucleus is the on the way, the nice, neat packaging in your trunk. So histones are the proteins that facilitate that. The DNA interacts with the histones and wraps around them, and then the histones can interact with each other and make kind of this second level folding, and it allows you to package the molecule very efficiently so that it all fits, right? So there's regions of the DNA that bind to the histones, and that's obviously very important. There's other stuff. So there's other like kind of stuff like this. There's like scaffolding attachment regions is the word you hear. It's DNA binding to proteins to facilitate the structure and the folding of the DNA itself, basically. So I lump all that stuff together under structural regions.
Now, is the packaging crucial to all of this working, meaning it has to be packaged a certain way to work?
It has to be packaged. So there's kind of there's two answers to that question. One is that you have to always be packaging some of it in order to let the rest work well enough, because if it's all just unpackaged, then it'll take up too much space and your proteins will be tripping over each other and they won't get where they need to go. So within the nucleus, you have two kind of general descriptors for the DNA. So the state we describe it in, just when your cell is just doing its normal day-to-day job, is called chromatin. And then the region that we call euchromatin is the less densely packaged DNA. So that's the stuff where you could have like active gene expression. And then the stuff that is more tightly packaged is called heterochromatin. And that is, going back a minute, tends to be methylated, and that facilitates the more dense packaging. And so that's the stuff that, like, I don't need this right now, just like go over there, don't be in the way. And these categories are fluid. So regions that are euchromatin at one time can be shut down and stored as heterochromatin at a different time. And you could take some heterochromatin off the shelf and open it up and use it later on. So these are not permanent things. It's fluid. within the cell. The other side of this, and there's newer research on this, so it's not as well documented, but there is research looking into the three-dimensional arrangement of all this stuff within the nucleus of the cell, and basically, like, is there like a card catalog system where, okay, this chromosome, when it's condensed, it sits over there in association with this other one, and they kind of interact and keep each other where they belong. So that's another way where you could look at this as very functionally important, right? This interaction with the histones and the other DNA is important in terms of just organizing the shop floor, if you will, just to keep everything clear and out of the way so that it doesn't get in the way and you can do the things you need to do with the stuff you need to use at any given time. That was probably more complicated of an answer than you were looking for.
That's fine. Thank you.
That is the nature of bio. Ask a simple question, get a complicated answer.
Got it.
So then there is a whole bunch of DNA that is non-protein coding, but it codes for RNA that actually does important jobs. So RNA is a molecule like DNA. It's ribonucleic acid as opposed to deoxyribonucleic acid. So it's a little different, but very similar structurally. And there's a bunch of categories of RNA that are really important. So ribosomal RNA, or rRNA, is actually going to form the catalytic core of this structure called the ribosome, which makes the polypeptides, it makes the proteins. So really need the ribosomal RNA, super important, even though it doesn't code for a protein. And then related to that, we have the tRNA, the transfer RNA. When you're making proteins, you have to bring in the amino acids. Those are the monomers that you're using to make your protein. The transfer RNA are teeny little pieces of RNA. They're only about 80 nucleotides long. It's a little single-stranded little piece that's folded up on itself. And to give you an idea, it's 80 nucleotides versus 3 billion for the haploid genome. So these are teeny, but you have a lot of copies of these genes in your genome. So there's a lot of them within your genome, but because they're so tiny, they make up a teeny fraction. But they're in there, and they're really important because they're the things responsible for, the term for the process is translation. When you're making proteins, it's the tRNA is actually doing the translating from like the genetic code to the amino acid sequence. They're the interface there. And then we have a couple other categories. We've got microRNAs or miRNAs. These are little bits of RNA that are often regulatory. Not all the time, but oftentimes they will interact with something that has a downstream effect. Like they'll interact with the mRNA I mentioned earlier during gene expression and affect the rate of translation, for example. And then one more, we've got long non-coding RNAs, I always say that. I've never actually heard this said out loud. I can't even remember, but I always say it in my head link RNA, but it's long non-coding RNAs. These are longer pieces of RNA that are comparable in length to like an mRNA, so they could be thousands of nucleotides long. And most of them are not going to do anything important, and we'll talk about why that is later on. But these can also be regulatory in that they can affect things like the rate of translation. Okay, so that's RNA that could be doing stuff. So you've got regions of the genome that code for the RNA that's doing whatever jobs it's doing.
Okay, so non-coding means non-protein coding, correct?
Correct, yeah.
Now, this will show my ignorance, but is that a good term to say non-coding? Is it not coding at all, or is it just not coding for a protein?
Right. And so that's actually, it's funny you said that, because as you were asking the question, I was thinking, I'm going to have to clarify this because not everyone uses the phrase that way. Yes. So oftentimes people will say non-protein coding to be more specific, to indicate it does, maybe it's transcribed, but it's not translated. Most of the time, conventionally, non-coding means non-protein coding, because when people talk of the genetic code, we're talking about using the letters in the DNA sequence to tell you the amino acid sequence, right? It's that changing of media from one molecule to the other. That is the code. But you'll also have people that will, for example, use the phrase RNA encoding genes. So this gets into the question of what is a gene. And the narrow kind of definition for gene is just a region that codes for a protein. But the more expansive definition for a gene is any region in the genome that has a heritable impact on phenotype, on a physical trait in some way. And not all of those regions are going to be protein coding, right? So there are, yeah, there's complexity in how people use the word. So when I say non-coding, I mean non-protein coding, but that is, and that's the typical usage, but it is not universal.
Okay.
Yeah, and
Thanks.
and the phrase that I'll often use is I'll say non-translated RNA or non-coding RNA or even just RNA gene to specify this region is translated into a functional RNA, but it's not, or sorry, it's transcribed into a functional RNA, but it's not further translated. It doesn't do the next step to a protein. Yeah. There's a lot of vocab in bio, and this is, my students have come to hate it when I say I have to start with vocabulary because they know we're just going to get like 10 words before we do anything fun. I just have to give them a bunch of definitions, and it's a good example of that. All right, and then all the stuff we've talked about is actually a minority of the genome. So there's tons of other stuff in there. So there are regions called pseudogenes. So these are regions that used to be protein coding genes, but are no longer protein coding genes. So for example, we have very famously in us, we have the gulo pseudogene, which is why we cannot make vitamin C. Other primates, other apes, they have the functional gene. They can't get scurvy because they can synthesize vitamin C. But right, you look in the same place in the genome where all these other primates have that functional gene. Ours is there, the sequence is there, but it's dead. It doesn't do the job. So we call regions like that pseudogenes. They were previously protein coding, now they're not.
Okay, let's stop here for a second. I have a ton of questions.
Yeah.
The term non-functional region, how are we defining functional versus non-functional? I feel like that's going to be super important.
We're gonna, don't worry, we'll get to it. So functional, the way that function is typically defined, and there's a very famous paper that defined it differently, which is why this gets confusing. But the way function is typically defined is something along the lines of promotes the fitness of the organism, affirmatively contributes to the physiology of the organism. And when I say fitness, I mean fitness in the evolutionary sense, not in the physical fitness sense. Fitness in the reproductive output in your population sense. So if the region of the genome is doing a job that basically helps you compete or helps you reproduce or survive in some way, then that region is functional. If it's not doing any of those things, then it's non-functional. And another way to think about this is biochemically, if you can remove the region and there's no cost to it, then it's non-functional. If you need it, then it's doing a job, right? That's the idea.
Okay. Yeah, that's my mind is going right now.
Yeah. Go for it. Just think out loud. That's why we're here.
Okay, great. So how are we able to say confidently that these were protein coding genes in the past? Like, what evidence do we have of that?
Yeah, so it depends on the pseudogene. But generally what we can see is that the sequence matches protein coding genes in other organisms, but something is wrong with it. So, for example, you need, in order to be a protein coding gene, you need a little marker called a start codon. It just says start building your protein from here. And if you have the start codon, you are what's called translationally competent. You can start building a protein at that site. If you don't have a start codon, you can't make the protein. So we can identify, for example, like the gene sequence might be 80, 90% the same as it is in some other organism, but we're missing the start codon. And then that gene is never going to be a polypeptide, right? It's still sitting there in the genome, but it's never going to generate a polypeptide. And we can look specifically for like the vitamin C example. We can look at like the human genome, and we can find it's got like this biochemical pathway where you take A, you turn it into B, C, D, and at the end you get vitamin C. Right at the end of this pathway, we have the whole pathway. It's all there, but one of the enzymes is gone, and we have a pseudogene instead. And so we can't actually do the pathway, but it's there. And again, specifically for humans, we can look in at like our specimens in the past, and we can look at diet, and we can look at things like nutritional deficiencies based on what we see in those specimens. And this is not my area of expertise, so I'm sure somebody in the world can explain this way better than me. But you can actually do the pathology on specimens that we find. And basically, we have evidence that we were able to synthesize vitamin C in the past, and then at some point we lost that ability.
Now, when you say that it's there, how many base pairs are we talking?
It's for the gulo pseudogene. Somebody in chat, I'm sure, knows better than I do, but it's like most of the gene is there. I think for that one we're missing. I think it's the promoter that we're missing, so it actually doesn't get transcribed. But like that whole gene, I can look it up real fast. Pardon the clicking on the computer, everybody. My keyboard's right by my mic here. Gulo pseudogene length. Let's see, because I actually don't know off the top of my head. It's 32 kilobases, so 32,000 nucleotides. 32,000.
Okay. And you're saying it's identical?
I wouldn't say identical, but it's going to be, in terms of its nucleotide sequence, it's going to be very similar to the functional genes that we find in other species. And not just primates, but most mammals can synthesize vitamin C.
Okay. And when we're saying that this is a non-functional region, we're saying it does nothing.
For that example, it pretty much does nothing because a lot of pseudogenes are actually transcribed to RNA and then not translated to a polypeptide. This example, because correct me if I'm wrong, chat, but I think the gulo example is actually missing the promoter. So it is not even transcribed to RNA. And then the other aspect of this, and Templar Warden in chat just beat me to it by like 10 seconds here, is that gene constrained? So this is a topic we're going to get to in a little bit. The answer is no. So, and that's an important question when we're evaluating function. So constrained refers to the presence or absence of purifying selection, which is selection that keeps the sequence the same, right? Selection against mutations that change the sequence. If a sequence is critical, you're going to keep that sequence the same, and if you mutate it, you're going to select against the mutated version. And we can detect that in the genome. Like there's regions that we can detect, say this one has very strong selection, this is very weak selection, this is the absence of selection. And one of the ways we can identify pseudogenes is they have the hallmarks, like structurally, of a gene, but they don't experience purifying selection. So they're building up mutations with no cost because they're not doing the job it looks like that sequence should be doing because it resembles a gene sequence, but it's able to freely mutate without cost, indicating it's not actually doing that job.
Okay. Let me ask another question. Again, I'm trying to wrap my mind around this, and it's not easy.
Yes.
So this is something that is in every human being.
Mm-hmm. Yep, we all have this region in our genome on chromosome 8, I want to say, and you could just go into the sequence, be like, boop, there it is. Gulo pseudogene is how it's annotated. Yeah.
Okay, so help me understand how we would get our entire population to have this. That doesn't make sense to me.
Yeah, so this gets into the pop gen of it. So what's going on here is this would be genetic drift, basically, although you could envision a situation where there is a selective benefit. And here's the idea. So for our history, our diet was fairly rich in foods that have vitamin C. And
can I stop you there?
Yeah, go ahead.
I have spent most of the last 15 years studying money and diet. So I have opinions on diet.
So
I don't think we can say across the board that everybody had a specific diet, because that's just false. Like
So
yeah, go ahead.
I agree that we can't say everyone had the same diet, but what we can say is that there was a transition at some point from a hunter-gatherer emphasis on gatherer diet to an agriculture starch-based diet.
Mhm.
And in that hunter-gatherer diet, that's going to involve enough vitamin C that you're not going to get scurvy.
Hmm.
Which me and you don't need a lot. The metric that I use for my class when we talk about this is the vitamin C in a single ketchup packet from McDonald's is enough vitamin C to not get scurvy. You have that minuscule little bit every day, and you're fine. That's all you need. So those like giant 1,000 milligram like vitamin C thick supplements you can get, yeah, you just pee all that stuff out. That's not staying in your system. So you only need a little bit of vitamin C, and a hunter-gatherer diet gets you there no problem because there's enough plant matter in there that's going to be something other than starch-based, where you're going to get the vitamin C. And if everyone's getting the vitamin C, if you have a mutation in that gene that inactivates it, there's no cost to that because you're getting it in your diet, so you don't need to synthesize it from non-vitamin C sources. So there's no cost to losing that gene. And there is an energetic cost to expressing a gene. So if you're in a situation, so there's two kind of hypotheses here. One, just genetic drift, random fluctuations in frequency, where just by chance the pseudogene won out. And that's just what happened to be what we all have. The other hypothesis is that because one gene is not going to save you a lot of energy, but it's not zero. And I'll actually talk about this a little bit. There's a paper from 2015 called The Bioenergetic Cost of a Gene. And basically, they found that there is a selectable cost associated with making a protein. And so if that protein is not helping you, then it's beneficial to not make that protein. The energy that you save is worth more than having the protein, basically. So the other hypothesis, specifically for the vitamin C example, is that it was beneficial because you got enough in your diet and it saved you that little bit of energy, and that was enough that if on net you had the pseudogene, you had more kids relative to the people who had the functional gene. So those are the two hypotheses. I tend to lean towards the drift hypothesis myself because the amount of energy from one gene being expressed or not is quite low. I would be surprised if that was a strong enough selective pressure to achieve fixation. But it's also not either or. It could be a combination of both.
Okay, I won't keep us here. I will say, though, just so people know what's going through my mind, is this sounds like the opposite of evolution to me. This sounds like the exact opposite of everything I've been learning from Erica, which is that these beneficial things are what end up taking over. And you guys are saying this is a negative thing that we had that everyone has lost. So I'm struggling with that a bit.
So evolution doesn't necessarily have a direction, right? Evolution is the benefit that we talk about with evolution is in terms of your fitness, which is your ability. It's your ability to contribute your genes to the next generation relative to the other members in your population, right? And if you have a trait that makes that number bigger for you and smaller for them, then that trait is going to increase in frequency. And so that is all going to be very situation specific, right? Fitness is not an absolute. It is a situational context dependent thing where in an environment where you have fruit, there's no cost to being unable to synthesize vitamin C. But in an environment where your diet is 99% starch, now there's going to be a cost to losing the gene to synthesize vitamin C. So it all depends on the relationship between the organism and the ecology, the environment, the selective kind of pressures that they're experiencing. And sometimes that leads to greater complexity, sometimes that leads to lesser complexity. It totally depends on the situation.
Okay, yeah. I can't remember who it is. I was listening to Jordan Peterson a long time ago on Joe Rogan, and he started talking about his daughter's diet, and I looked into that. And there was a people group in the past that lived in a very cold environment, probably north of the Arctic Circle, would be my guess. And I'm pretty sure they didn't have vitamin C in their diet at all. I think that they were 100% animal based.
Possible. I I don't know that specific example, so I
Yeah.
can't comment on it.
Yeah. I don't know enough either.
I would be skeptical of anything I hear from Jordan Peterson via Joe Rogan. Let's put it that way.
Okay.
When
Yeah.
when Jordan Peterson speaks on fields that I am familiar with, it causes me physical pain. He
Okay.
is when he speaks on the things I am familiar with, it's way off base. So I would be just skeptical of taking things at face value without double checking them.
For sure.
Yeah.
For sure. Okay. All right. Let's keep going.
Let's keep going. Okay. Other regions of the genome, origins of replication. So you have, by the way, Chad is saying you get vitamin C from animals, which I'm not a nutritionist, but sure. Okay.
Well, then they tell you that people got scurvy from eating meat. So
It has to be fresh. I don't know. So, fresh or I don't know. Okay. Origins of replication. Basically, you have to copy your DNA. The regions that say start copying here, you've got a bunch of those copies sprinkled throughout the genome. So that's an important thing. It's not a lot, but it's not nothing. Then we have a ton of these things called mobile genetic elements, which fall into a bunch of different categories. So you've got retrotransposons, endogenous retroviruses called ERVs, and then DNA transposons. So what the heck are all these things? So these are basically different types of genomic parasites. Retrotransposons do this copy and paste thing where their RNA, they use an enzyme called reverse transcriptase to, instead of going DNA to RNA like regular transcription, they go RNA to DNA, and then they cut your DNA and stick themselves in there. And now you've got this little extra bit in your genome, and then that little extra bit can be transcribed, and then it makes another DNA copy and sticks itself somewhere else. So it does like this copy and paste thing. So we've got these retrotransposons sprinkled throughout your genome. Then we have DNA transposons. I'll do those next because those are similar, except those are cut and paste rather than copy and paste. So you have a lot fewer of those because they don't propagate as rapidly. But these are regions where it's a bit of DNA, it cuts your genome, puts itself in there, and then it can cut it out and go somewhere else. And then endogenous retroviruses are a really interesting one. These are the remnants of viruses, where you have a virus called a retrovirus. This is like HIV is a retrovirus. So the virus is RNA in the viral genome rather than DNA, makes a DNA copy, and inserts the DNA into your genome. And then we actually have, our immune systems actually have ways of recognizing retroviruses and target those regions to mutate them so the virus can't propagate. So it can't then, what we say is it can't mobilize, it can't come back out and function as a virus that's then going to kill your cells. So we have a bunch of these viruses, once they get into your genome and then they inactivate, either through the activity of our enzymes targeting them or just random mutations and they get broken, we call that process endogenization because now they're stuck in your genome. They put themselves in as a parasite and now they break in some way and now they're stuck there, they can't get back out. So now we just have like this dead virus taking up space in our DNA. So those collectively, we call those things mobile genetic elements. And here's a fun thing for you. I see this comment here. Mine doesn't. Stupid shingles. That's different. Those viruses, that's the chickenpox virus, those viruses are not integrating. That's not a retrovirus. Those viruses actually go into a resting state. Sorry, Will, this is totally off topic, but it was fun. Those viruses, the chickenpox virus, goes into a resting state in the basal root ganglia of your spine. So they're not integrating into your genome. They're just dormant for decades. And then at some point later on, they boot up, they travel down the axon of those neurons, and that's why you get the rash on one side and it's at the periphery, it's because it traveled from your spine outwards to the periphery and then it does its thing on your skin. So that's actually a different thing. That's not a mobile retrovirus. That is a different thing. There you go.
I've never looked into endogenous retroviruses, but I hear Donnie Badinsky talking about them a lot.
Yeah,
So I'll have to go hear what he has to say.
I have his book on it. I reviewed
Oh,
his book on it. He's way off base on it. I'll just give you the preview. He'll say that they're not viral in origin. They were created in place as functional elements. But we know that they're viral in origin because, in addition to having all the hallmarks of a virus that I'll describe later on, they have what's called a target site duplication, which when the virus inserts itself, if you've got like AATT in your genome where it inserts, if you have no virus, it'll be blah, 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 AATT, blah, 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 blah. When the virus inserts, it's blah, 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 AATT, the virus, AATT again, and then the other stuff. That's called a target site duplication. And we can actually directly show that those are the result of viral integration because we have examples of retroviruses that are active in humans, in koalas, and a bunch of other things. And you can look at an individual who has the insertion at a particular site and you see the duplication. And you have the individual without the insertion, no duplication. So the duplication is actually the result of biochemically how the insertion happens. And so the target site duplications, in addition to all the other evidence that they're viruses, the target site duplication is just dead to rights. That is a viral insertion site. We can, like, we have directly observed the mechanism of viral integration. And so we can spot these sites in our genome. It's like just once you know what to look for, it's just a giant spotlight on them. And there's about 200,000 of them in the human genome. About 200,000 ERV insertion sites. And the target site duplication is like the telltale, this is a viral insertion site.
okay, great. There's someone in the comments, Azaria, is asking me why I would go listen to Donnie about this, which is a very bizarre question to me to ask. Because imagine a creationist said, "Why go listen to Dr. Dan?" That you'd immediately jump on that.
To be fair, you might, for a topic that's relevant for biology, evolutionary biology, you would listen to a biologist.
I would listen to anyone who has studied it and has an opinion. I'm sure you remember the bar scene from Good Will Hunting.
Remember it well.
That's my favorite scene of a movie of all time. So the idea that you have to have a PhD to have truth is just false.
Well, we're not here to talk about Donnie.
Okay.
But after you talk to him about herbs, watch my review of his book. There's some basic
for sure.
problems there.
I just want to make sure I'm hearing everything
Yes.
and make the decision after that.
All right. So that's what's in the genome. So now we can get to what does it all do? So this is where we're going to try to figure out what percentage is functional. And anything that's non-functional, that's the junk. And again, non-functional we're describing as stuff that doesn't help you survive affirmatively in some way, right? If it's doing a job that helps you, it's functional. If it's not doing that, it's non-functional. So right off the bat, the expectations, the prediction is that we're going to have a lot of junk DNA. And there's a bunch of reasons for this. So I'm going to go through 3 reasons why we expect a lot of junk DNA here. So first is something we mentioned a minute ago. Whoever asked in the chat beat me to it. Thank you. Most of the genome is what we call unconstrained. So this is a study from 2014, and they found that 8.2% of the human genome experiences purifying selection, which is to say, if you change the nucleotide sequence, there is a cost to that, and you're going to select against those mutations. So basically, if you change it on net, you're going to have less reproductive output than if you keep it the same. So that means that over time, the sequence that is optimal, the one that works, you're going to keep that the same over time. That's called purifying selection. And that only applies to between 8 and 9% of the human genome. So we only need the sequence to be the same for 8-ish, 8.5% of the genome. And most of the functions we're going to talk about, almost all of them require specific sequences, which means they're all going to be completely contained within that 8%. Right? So that's the first reason we expect a lot of junk, is that very little of the genome actually, the sequence, doesn't actually matter for the vast majority of it. The second thing is that there is a very low cost to having a bunch of DNA that's not helping you, as long as it's not hurting you. So this is a 2015 paper called The Bioenergetic Cost of a Gene. And again, I'll recommend Dr. Zach Hancock on this, because he goes into the actual like math of the population genetics here. But basically, within a population, you're going to have differences in fitness between individuals that are too small to matter in terms of their relative success in passing on their genes. There's just going to be little differences that are so small they don't matter, and that difference, we say, is below the selection threshold. And what this paper did is it looked at the cost of having a gene, or having a region in your genome and replicating it, transcribing that region to RNA, and then translating that region to protein. So there's kind of three things that you have to spend energy on: copying the DNA, making the RNA, and making the protein. And if you do the second or third, that implies that you're doing the ones below it. So if you're making the protein, you're also making the RNA and copying it, right? It's kind of a cumulative thing there. And what they found is that for I forget if it was specifically for humans or mammal populations, but for like natural populations out in the world, the cost of making a protein that is non-functional is generally high enough that you can select against it. But the cost of making the RNA, as long as you don't do it too much, and the cost of keeping that region in your genome and copying it every time you copy your DNA, that cost is actually too low to be selectable. So you have, for example, an endogenous retrovirus, say, a virus that's not doing anything, it's just sitting there, it's broken, it's just taking up space. The cost of having it versus not is so minuscule that there's no selection against it. Which means, because we have mechanisms to add stuff to the genome, but selection is not going to remove it if it's below that threshold, we expect over time a bunch of this non-functional stuff to build up, right? So that's the expectation, just based on the kind of evolutionary genetics aspect of it. We expect a significant amount of non-functional DNA to be present based on what we would calculate you could remove via selection and what you would kind of be stuck with, right? And then the third one, which I think is not the best argument, but it's the most fun. It might be the best argument, but it's certainly the most fun, is called the C-value paradox. And a kind of a variation of this is the onion test. Have you ever heard either of those terms?
Mm-hmm.
Yeah. So the C-value paradox refers to wide fluctuations in genome size across lots of organisms that's uncorrelated with things like how many protein coding genes do you have, how big are you, how complex are you in terms of your development or different tissue types that you have. And it's also within like specific taxa. So, and this is where the term the onion test comes from. An onion has a genome, diploid genome, that is 30 billion base pairs, as opposed to the human diploid genome, which is 6 billion base pairs. Other members of that same, I think, family for the onion, I'm not a plant biologist, I don't know what these groups are, but within the same family that onions are found, you find an order of magnitude difference in there, where you've got things all the way down in, comparable to like the human-sized genome in the single-digit billions, up 10, 15, 20, up to 30 billion for the onion. These are very similar organisms with vastly different genome sizes. Very similar in terms of their numbers of genes, very similar in terms of their physiology, number of tissues, all that stuff, but vastly different genome sizes. So that's the onion test, which again is related to the C-value paradox. And the idea with the onion test is that it's kind of like a little self-check here. Like, okay, if the hypothesis is that the genome is mostly going to be functional, then we need an explanation for why the onion has all that DNA that's mostly functional, but a very similar plant has 20% of that DNA. what's all that extra functional DNA doing in the onion that the other plant doesn't need, despite them being physiologically very similar. And we could expand this out to even more extreme cases. So you'll notice in this graph right here, so the bars, this is probably too small for everybody to read. I apologize for that. This is not a high-res figure here. But the bars, by the way, this is from Larry Moran's blog, is where I got this picture. So blame him for the resolution. But this, the bar represents the range of genome size. And across the axis here, it's a logarithmic scale. So we've got 10 to the third, 10 to the second, 10, 1, -1, and so on. And you'll notice this really long bar here, protozoa, those are unicellular eukaryotes. So the biggest genomes all the way out here are actually unicellular amoeba. And their genomes are 500 billion base pairs long. It's in the 500 billion range. And again, humans are 6 billion for diploid genome. So having a unicellular organism, which obviously has much less physiological and developmental complexity, having that enormous genome, if we're going to posit that the genome is mostly functional, we have to have an explanation for why that's the case, right? So that's the C-value paradox. So these factors together lead to a prediction of most of the genome is probably non-functional. That's the conclusion to draw from these observations.
Okay, let me ask a question.
Yeah, go ahead.
I personally think that predictions are super important in the world of science. Based on what you're telling me, let's stick with the C-value paradox for a second. Can you guys make predictions concerning the C-value paradox prior to us sequencing genomes?
So we could have a rough estimate of the number of protein coding genes in a genome based on like biochemical assays for genes. So what you do is you take a cell type and you isolate all the proteins, and you do this, it's called a two-dimensional gel, and you get like, it's like a scatter plot where each point physically represents a protein that you've isolated, and you're comparing it across like size and like other biochemical properties, so you separate them. And so you can get an idea of the
But
number
I'm not
of
talking about genes. Like the number you said for the onion, what was the number?
for onion, it was 30 billion nucleotides.
So I'm talking about nucleotides.
Yes. You could do it by weight prior to actually sequencing. You could actually do it and just show that like this cell has more DNA than this other cell. So prior to sequencing, you could show that there were big differences there, but we couldn't do it precisely until we could actually sequence the genomes.
Okay. Now I don't know the answer to this, but I'm trying to put the connection in my brain is I feel like for what you're telling me here, well, let me ask you this to make sure I'm not messing this up. Is the C-value paradox in your mind coming from time? So the greater the value, the greater the time.
I don't think so. I'm not sure exactly what you mean, but I don't think so. So nothing about this has anything to do with like the time to most recent common ancestor or anything like that. This is all just observations in the present based on just what we can see about the genomes.
Okay, so what does, I don't know what to call it. It's terrible. Okay, I'll call it mainstream science. I think that would be good. What does mainstream science say as to why the onion has such a large genome?
Is that you have mechanisms that lead to an increase in the size of the genome, and then that additional size does not carry a selectable cost with it. So having the larger genome size does not come with a deficit in terms of like how much energy you need to spend copying it or whatever, right? That the difference in having the really big genome versus the not as big genome is not selectable. So you have mechanisms like little duplications within the chromosome, for example, that'll increase the size of the genome over time. But then once that happens, there's no selective pressure to get rid of it, right? If an individual happens to experience a deletion that reverses it, there's no benefit to that versus having the duplication in the first place. So I don't want to call it ratchet-like because eventually you do run up against constraints, but to an extent, there is a ratchet-like increase in genome size with limits.
Okay, and so this can happen in a short period of time, this increase?
You can actually have it in plants in particular, yes, through a mechanism called polyploidy, where you can have a full genome duplication, and then you double everything. And so a lot of plants are, so we talked earlier about haploid, which is one copy of everything, diploid is two copies. A lot of plants are either tetraploid, hexaploid, or octaploid. So that's 4, 6, or 8 copies of everything. And what tends to happen is that you have these genome duplications, and then because you have multiple copies of each gene, if one of them mutates and becomes non-functional, there's no cost to that. So you end up with this genome that is littered with broken genes because there's no cost to having the extra DNA, and you had kind of a backup copy of the gene, right? Because you had these extra copies. So you end up with a bunch of DNA that's not doing what it did in the past, but it's still taking up space. And we can do this, to give you an idea of the timing of this, you can do this as somebody that I've spoken with will explain. You could do this in your garden over a summer. You could get, and I forget the 2 species, but it's like you could get this species, and they hybridize really easily. And when they hybridize, they become tetraploid. And then that hybrid is a totally different thing. So you can do this very quickly and like get that genome to get a lot larger.
Okay. Okay, I'll have to think about this one. It's a little confusing, which is probably why it's called a paradox.
It's a lot. Yeah, it's a lot. Any questions about the constrained or the cost aspects there?
The cost one is interesting to me. It seems a bit arbitrary, but maybe I just don't understand it fully.
It's so that would, to really get into that, we'd have to like take out a whiteboard and do population genetics math, because the relevant measure is this thing called effective population size, which is basically how big does your population have to be to get a representative like sample of everyone reproducing in that population. And that dictates the differences between individuals that are selectable in terms of how well they compete. And the bigger the population, the smaller the differences you can select. So in very large populations, there are situations where you could select for a smaller genome very efficiently because those small differences are detectable. But in a smaller effective population, like a mammalian population tends to be pretty small, compared to like bacteria or, fish or something, you tend to need larger differences in order to make it selectable. So there's math under the hood there that it would take an hour to walk through all that, and we just don't have time.
That's fair. Let me ask you one more. Let me ask you one more time on this term non-functional. That to me seems like it would be a very difficult thing to establish with certainty.
Yes, it is very difficult to establish with certainty. It is relatively straightforward to establish with high confidence. Not certainty, because I am the first one to tell you there is almost nothing in bio that we can establish with certainty. One thing I like to convey to my students is physics is a saying, and I forget where I heard this. I apologize, whoever I stole this from. I forget where I heard it. Is physics has laws, chemistry has rules, and biology has exceptions. So things work 90% of the time, 95%, 99% of the time, they're a certain way. You can almost always find some weird thing that doesn't work that way in bio. So we can establish, and there's criteria that we'll use to establish what makes this thing functional versus not. And there's a paper from 2014 that does a really nice job laying it out. But yeah, I would never like to use the word certain. But we can definitely establish things with reasonably high confidence. Given if a region of the genome meets certain criteria, we can be pretty sure that it's not functional. Right?
What's that criteria?
We'll get there. That's on slide.
Okay. Perfect.
We'll get there. It's all the way through all that. We'll get there. Don't worry. I promise we'll get there.
All right.
Okay.
Thanks.
So now let's do the fun part. Let's do the percentages, right? Because the question here is, we're talking about junk DNA. Is it real or is it not? So let's just go through all the parts of the genome and what can we assign function to or not, right? So we're going to do this region, percent, region, percent, region, percent. And the numbers I'm going to give, I'm rounding up very generously here because some of these are like 0.1%, and we're going to call it 1%, right? We're gonna round nicely here. So exons, are the first one. So those are those protein coding regions that actually tell you what amino acids to use when you're making a protein. Those account for about 1.5% of the genome. Everyone agrees exons are functional. Like, they're just, obviously exons are doing an important job, right? Then we have those regulatory regions I mentioned, promoters, enhancers, silencers. There's a lot of variation in the estimates for these, but the consensus is in the high single digits, mid to high single digits. So 7 to 8% is generally what you're going to find for those regions where they're not the protein coding genes, but they're the little regions that will affect the activity of those genes, right? So 7-ish% there.
Are these numbers coming from Encode?
These are coming from Encode and a bunch of other places. There is a table right here. This book was from 2024, I want to say. But yeah, Encode will have a similar thing in their various papers.
Okay.
This book was published in 2023, and we've got on page, I'll tell you. Here we go. It's on page 133. You have this table. I'll hold it up for everybody there. There you go. I got a table, and it kind of breaks it down percentage-wise there. Now, my numbers are going to be a little bit different from what's shown in that table for two reasons. One, I group things a little bit differently, and two, I'm a little more generous in terms of how large the numbers can be for certain categories. So the numbers are going to be a little bit different between what's on screen here and what's in the book, but they're comparable. And then we have, I mentioned the origins of replication. We'll call that 0.5% to make the math easy. It's actually like, I think it's like maybe 0.3%, but we'll call it half. And then we've got the functional RNAs that we talked about, ribosomal RNA, tRNA. You lump together all what you might call RNA genes that have a directly documented role to play, and that gets you about 2%. And then one more, we've got those structural regions. So you've got your centromeres, your telomeres, your spacers, like the histone binding, like all that stuff. That gets you approximately another 3% of the genome. So you add all this stuff together, 3 and 2 and 7 and another 2, and that gets you about 14% of the genome. So that is higher than normally what you hear for this. If you ask some random biologist, like, who cares about the genome and what's in it, what percent is functional, the number you're going to get, or let me rephrase that, what percent have we like documented a clear function for? The number you're going to get is usually somewhere in the neighborhood of 8 to 10%. We're getting 14 because I'm being a little generous with, in particular, the structural regions. Origins are a little high, promoters and such are a little high, and exons, it's like closer to 1, but we'll call it 1.5. It's between 1 and 2, but probably on the low end of there. So we're going to call it off the top just like 14% of the genome. Everybody can agree generally that that 14% of the genome has a directly documented function. No controversy there. There's other stuff that there's controversy about, but this part, we can just put that down. Okay. So what about the rest of the genome? All that other stuff we talked about. All right, so we've got retrotransposons, those copy and paste little parasites. 45% of your genome is retrotransposons. 45%. Which makes sense because they're really good at copying themselves. That is their job, right? All they do is, hey, I'm here, I'm going to make a copy, I'm going to put myself there. I'm going to put myself over there. So our genome is just littered with these retrotransposon sequences. Then we have the DNA transposons, the cut and paste ones. They're not as good at propagating, so that's only about 3%. Then we have the endogenous retroviruses, the remnants of those viruses that invaded. That gets you about 8%. And that's, I want to be clear for everybody, that 8% is just the ERV stuff. That's not including like the genes that we have co-opted. That's in the exon fraction that we already talked about, right? And then we have pseudogenes. That's about 5% is pseudogenes, right? Then we have a whole bunch that is hard to classify as any one thing. It's unconstrained, non-repetitive. So basically, it's regions of the genome where the sequence doesn't matter and it's not just a bunch of repeats. A lot of the rest of the genome, like a lot of the transposon stuff, is repeats because they have these telltale sequences. Whereas this unconstrained non-repetitive DNA is just, it doesn't experience purifying selection, so there's no cost to mutations occurring there. It's non-repetitive, which is to say it's not like the same 3 or 5 or 10 nucleotides over and over again. It just sits there and it doesn't have like a ton of biochemical activity that it's doing. And most of the time it's going to be, going back to what we talked about earlier, it's going to be condensed into that heterochromatin. So it's not even really like available to do stuff in the cell, right? So that's about 20% of the genome is that unconstrained non-repetitive DNA. So you add all that stuff up and that gets you about 81% of the genome in these categories. And now just to like be clear for everybody, we're just describing how much space this stuff takes up. We're not right now wading into the is it functional or not. At first pass, this stuff appears to be non-functional and it adds up to 81% of the genome. So our running total is 14% functional, 81% junk. Now you'll notice that doesn't add up to 100%. That's because some fraction of the genome, even now, is not well classified. We can't even put it in any one of these categories. So we're just going to like not worry about that stuff. I know it doesn't add up to 100%. We're just going to not worry about it. There's other stuff. It's, what is that, like 4%? So 5%. So it's not much that we're missing. But we've classified about 95% of what goes on in the genome on the last 2 slides here.
Okay. I don't know if this is the right time to talk about it, but
go for it.
The first thing I want to know is why Encode originally, I forget what year it was it 2012?
2012, yep.
Why did they come out and state that 80% was functional if they were all, credentialed scientists?
Because they used a different, and I will say idiosyncratic definition of function. They defined function as any biochemical activity beyond DNA replication. So any transcription, any association with a protein, any biochemical modification like methylation or acetylation, if they could assign any of those events to any part of the genome, they said that's function, right? So they're using a biochemical activity definition of function rather than a physiological definition of function.
Okay. And can you help me understand, I do not know where Encode got the genome that they sequenced?
I'm not sure what, and then probably, I can't say this for sure, but I would think they were using the human reference genome, which is like kind of the master human genome. And then they were looking at different cell types in culture when they were doing the biochemical assays. So basically what they were doing is they were using different human cell types, and I forget off the top of my head what cell types, but it's in the supplements to their papers. And they're saying, okay, within these cell types, we can identify these regions that are doing these activities, and then they're taking that information and annotating the reference genome according to, okay, this region has this activity, this has that activity, so on. Right, so that's what they're doing. They're using cell cultures to biochemically assay for activity, and then using what they find to annotate the kind of master human genome.
Okay, I like to just think about things. I don't mind being stuck in traffic because I can just think,
Right.
And something I was thinking about on a recent plane ride, it was a long one. I actually got separated from my family on the plane, but that was fine. I helped a couple with a newborn sit together, so that was more important. So I had a lot of time to think. And I didn't have Wi-Fi, so I couldn't do any research. But I was wondering, because let me get something out that I think is super important, and this is going to probably be foreign to a lot of your audience. But humans have a high opinion of themselves. Okay. We all do.
Yes, we do.
Okay. Including myself. And when we start talking about the world, um, I am extremely confident that God exists. So that's a filter that everything runs through in my life. And when we look at this world that I believe was created and designed by God, as smart as we are, we'll never even scratch the surface of the mind of God. And so I was thinking about Encode and I was thinking about junk DNA. And I was wondering if it's possible that certain portions of our genome are only active at certain ages. And that's why I asked you where they got what the genome
Got it.
that they sequenced. And I don't know if this has ever been tested, but that to me is something that God is absolutely capable of. And so if we see something that's not active, it could just be the person's age.
So, I will say that has been investigated. So you're looking at like developmental regulatory stuff, for example. You're looking at things that are only active during embryonic development and then shut down after. There are things in that category, and I'm going to, in a little bit, talk about how we can be confident that most of the genome is not doing that. So I'll get there. Before we do, I want to highlight 3 comments here from various people because I think they contribute to this conversation a bit. So first, from Joseph D. Zugavili, 7231. It Encode, this is referencing, was looking for things to study. If it lit up under Encode, it might be worth looking at. So that's a pretty good way to describe it. It's like a first pass of like, what's all the biochemistry that's happening here? Obviously, anything that's functional is going to be biochemically active, so that's where to look further. And so the 2012 paper was saying, here's all the activity. And then in subsequent publications, very famously a 2014 paper, they went more in depth into what that activity is and kind of categorizing it and describing what we would look for in that activity if we're going to attribute function and if we're going to attribute non-function within those regions. And I'll mention that paper later on. There's another one here from Fits of Rage 23, gum on shoe analogy incoming. Yeah, so this is a very common analogy when we talk about the different definitions of function. So the analogy is this, as what's the function of gum, right? And typically you would say to chew it, to enjoy the taste, right? Whatever, right? It has something to do with being in your mouth is what the function of gum is. Well, gum, when it gets on the sidewalk, it can stick to the underside of your shoe, and then it does that annoying thing where one foot is like sticky for, while you're walking for like half a mile until it kind of either comes off or gets sufficiently dirty that it's not sticky anymore. So by Encode's definition of function, one of the functions of gum is to make your shoe sticky, right? Even though that's not like what gum is for, right? That's not what you would say it was invented to do, but that's one of the things it does. Therefore, it would be attributed as a function of it, right? That's the difference between Encode's definition of function and the what you might call the physiological definition of function. And then one more from Mr. Anderson Crosses. They define function as whether it made the cell do stuff. And I would even just say that like, did anything happen in the cell, right? Rather than whether the stuff it made the cell do actually had any effect on survival. Right. So it's what are the things that are happening, not what are the effects of those things that are happening, because biochemistry is noisy and messy and a lot of it doesn't actually do anything. And that's an important thing to consider when we're trying to evaluate function specifically.
Yeah, so that last comment, I have to disagree. I can't define function as having an effect on survival. That I'm not willing to go there. That doesn't seem fair to me.
I would even say having an effect on survival would be too broad because there are clearly non-functional regions that have an effect on survival. So this is something you hear sometimes where you have a region that is inactive. So most of the time it's methylated, it's not being expressed in any way. If it escapes that control, it becomes active, and then that region causes a disease. So you can have regions that our cells regulate them to basically be like, stop, you part of the genome, don't do anything. And if that part of the genome does something, you get sick from it. So that has an effect on survival, but it's a bad one. So that region is not functional. That's not helping you, right?
Mm-hmm.
And I think that's an instructive thing to consider, that there are regions that are inactive, that when they activate, it causes disease. And that's evidence that that region is not functional rather than evidence that it is.
Yeah, I guess maybe I might be thinking of it too simplistically, but let me just give you an example off the top of my head. My beard, okay? My beard does not in any way help me survive. Yet it's there for a reason, and so I wouldn't look at code that is reason I have a beard and say it's non-functional. That doesn't make sense to me.
Okay, right. So in that case, though, if you reduce it to like the biochemistry of a cell is what we're talking about, though. So you're
Mm-hmm.
making molecules having specific interactions, right? Also, we
Mm-hmm.
have to, separate humans from cells in that, like, my beard doesn't do any function except I like having a beard. And, like, so that's
Mm-hmm.
my frontal lobe doing a thing. That's not like a biological, urge or anything to have a beard. It's just like, I prefer to have a beard. Uh, yeah, so I think we just need to separate that out from when we're looking at DNA in a genome and the proximate biochemical effects of what that DNA does or doesn't do.
Okay. Okay. All right, we can keep going.
Let's keep going. Okay, so all that stuff we just talked about, can we add functions? Because we tentatively said that stuff is non-functional based on what we know it is. We can tentatively say non-functional, but some of it has important functions. In fact, we have directly documented functions in every class of that stuff that we just described. Transposons, ERVs, all that stuff, we have directly documented functions in those regions. So we can shift our distribution a little bit away from junk towards function based on the stuff in those regions, okay? Now, the functions that we've identified, that should say regular, that should say regulate. That's what I get for doing these and not proofreading them. So it should be regulate gene expression via protein binding. So you have regions of the genome where proteins will bind, and that will affect the gene expression in the nearby region. And those regions can be from transposons or ERVs or whatever, right? So that's regulation. That's important. You can regulate gene expression via RNA. So you make a little piece of RNA, and that interacts with the mRNA before you make a protein. That's going to affect the rate of translation in some way. Or something else grabs onto that long non-coding RNA that would have otherwise grabbed onto the mRNA, and that affects the rate of translation in some way. We can call that stuff functional. There's a longer conversation to be had about like exactly how you split those hairs, but for tonight we could just say if it does that thing, call it functional. That's fine. And then another one that you see, particularly for endogenous retroviruses, is the possibility that they could fight viral infections. So if you have RNA from a virus and you have complementary RNA from your genome, from a virus in your genome that looks like that virus, they can match up, and now that's going to prevent that virus from replicating in your cells. So you can have regions in your genome that when you express them as RNA, that could actually help you fight viral infections. So these are all things that are found in those regions we just described, the transposons or the endogenous retroviruses, right? So here's examples of functions that can be found in those regions, and we've directly documented this stuff in those regions. So we cannot say that the entirety of all that stuff is non-functional. We have to say some fraction of that stuff has a function. Here are the functions we've documented. So we have to shift our numbers a little bit, but only a little bit.
By the way, people in the chat are trying to argue that a beard actually does increase fitness.
It's just an analogy, folks. It's just an analogy.
I do want to ask all those people that are making these arguments, like, being attractive or keeping me warm if it's cold. I want to know why women don't have beards. So.
It's just an analogy. we don't have to like belabor every analogy that's made. It's okay. All right. So, what are the functions we can add? So, want to be clear, in every one of those classes, we can assign functions. We could say, yeah, here's an example that does this specific thing, but it's very few, like vanishingly few. We're talking, in some cases, like single or double digits, like very, very few. And so that gets us more. So we'll just put all these numbers out. For retrotransposons, DNA transposons, ERVs, and pseudogenes, we have documented members of each class that are functional, but it is less than 1% of the overall genome in each class. In some cases, it's like a handful. Like for ERVs, it's like fewer than 20, but it's not zero. So what we're going to do is we're going to add 1% for each of these. It's way less than 1%. It's like 0.1% or 0.100%, but we're just going to add 1% for each of them. We're going to add 4% to our functional fraction, and that brings our running total. We're shifting 4% over, so now it's 18 to 77. And I want to say here that 18% number is way bigger than kind of the consensus range, which again is 8 to 10, maybe to 12. Very few people will land on 18. Generally, people will say it's around 10 or 80 to 90. Very few people are going to land in the 20% range. But because we're kind of puffing up the numbers a little bit, we're landing in that kind of no man's land. But that's a reasonable place to land based on kind of the level of detail we're going into here. Okay. Okay?
Yes. Yes, for now.
Okay. So now let's talk about that remaining 77% and how we're not just saying, we don't know, right? Let's talk about why I'm very confident it's junk and not certain, because again, it's bio. Never say never. We find weird stuff in bio that should not exist. There's some stuff we found that should not exist, but it does. So let's go through what's left. Now, most of the stuff that's left is remnants of either mobile genetic elements or genes. And I say remnants because most of them are actually not complete. They're degraded. They're missing like parts. It's not just that the sequence is there, but there are a few mutations. It's like part of what makes it a retrovirus, for example, is just absent. It's been lost over time. And in addition to that, there are lots of mutations that cause the sequence to deviate from, for example, the ancestral retrovirus. Like there's an example, I forget the name of it off the top of my head, but there's a retrovirus that we've identified that we've actually resurrected the functional virus. So you can take the retrovirus that's mutated and you can look at viruses that exist and say, what was the sequence of that probably when it was functional? And you could basically resurrect this dead virus. And we've shown in the lab that you can do that. So we know that there's mutations that have broken it, in effect, and made it not do the virus stuff that it would have done. And then the other aspect of most of these regions, in fact, all of the stuff we're talking about at this point, is that it's unconstrained. So earlier I said about 8 to 9% of the genome is subject to purifying selection. That means the actual sequence matters for whatever job it's doing in your cells. If it's unconstrained, the sequence itself doesn't matter. It can mutate freely without a cost. All of the stuff we're talking about now is unconstrained. There's no purifying selection, so the sequence doesn't matter. So you put this stuff together, all of that stuff that's left, we can very confidently say it can't do the functions we've talked about so far. Because those functions require specific sequences, with the exception of like the spacer DNA, where the distance matters, but the sequence doesn't. Other than that, all that other stuff we've talked about, regulating gene expression, protein coding genes, interfering with viruses, right? All that stuff, you need to have a specific sequence in order to accomplish that function, right? If you're, for example, going to interact with and interfere with a viral genome when it gets into your cells, the sequence in your DNA has to match the viral DNA. If it doesn't match, they won't interact, and then it won't do that function. So you need to have selection to maintain the sequence, to keep it the same, to select against mutations that would cause it to deviate, right? all the stuff we're talking about now is in that no purifying selection box, which means we can very confidently say it's not doing a function that requires a specific sequence. Does that make sense?
It does a little bit. I want to go back to my point about us being confident that there is no function. Another thing I thought of, so there's the age piece, which I still want to look into more, which I think is
And we'll
fascinating.
that was the next thing I was going to get back to, so don't worry, we'll get there.
Okay. And I'm not only talking about embryonic either. So all throughout your entire life. The second thing I want to talk about is environmental aspects. And again, I'm purely just like trying to think outside the box here. I'm actually coming, again, maybe I'm wrong, but I'm coming at this from a perspective of, if God designed this, why are we seeing, why do people think that there's no function here? And so I'm just kind of running through checking the boxes. Could it be that certain things will only become active in certain environments? Could it be that certain things only become active if you have an injury? Okay.
So yeah,
I don't know much about epigenetics.
That was exactly the note that I made while you were talking. Is yeah, now we're getting into epigenetics. So let me address both of these as best I can here. Actually, let me just see what I have next. Okay, yeah, we'll get okay, we'll get there. We're gonna go through this stuff in more detail, but yes, the so for the age thing, all of that and the environment, we kind of lump all this stuff together. Anything that's going to regulate development, whether it's embryonic or during your life, or things like changes to your metabolic genes. We know that your metabolic profile changes over the course of your life, right? Changes like that are all going to be regulated with sequences that are sequence dependent. They need to maintain a specific nucleotide sequence in order to be responsive to the signals to do that job. And when we're talking about responsiveness to environment, that gets into epigenetic regulation, where you're regulating gene expression patterns via methylation that we talked about before. So people will often treat epigenetics as kind of a separate inheritance mechanism from like classical genetics, where it's the sequence you're inheriting, because with epigenetics, what can actually be passed from one generation to the next is the methylation pattern, right? The kind of biochemical profile of the DNA, in addition to the sequence itself. Now, the thing is, that's not a separate mechanism. It's just kind of 1 degree removed from it, because those modifications of the DNA are carried out by enzymes. Enzymes are proteins that have a specific sequence, and the target sites in the genome where the methyl groups are added are also sequence specific. So you have to have a particular sequence at the target sites in order to be able to methylate the DNA. And if you don't have those sequences, then the methylation doesn't occur. So you can have these kind of, a lot of times like metabolic stuff, where if you're in a high calorie or low calorie environment, that's going to lead to a different gene expression profile based on epigenetic modification of the DNA. But that's still, at the end of the day, going to be dependent on having specific sequences that allow you to do that. So we can still exclude that non-constrained part of the genome from participating in that regulation.
Okay.
So let's go through these in a little more detail, each one. So the first thing is, you brought up Encode before. Perfect. 2012 paper. The first thing I want to we talked about this a little bit, so we don't have to belabor the point. But activity is not the same thing as function. And this is like, everyone cites the 2012 paper because they documented biochemical activity other than replication, because that would be 100%. Other than replication, they documented activity for 80.4% of the genome. And that could be transcription, so you're making RNA. It could be protein binding, chemical modification, any of that stuff. Any of that stuff gets lumped in under activity. Now, the problem with this is that biochemistry is super messy. A lot of these interactions are stochastic, and we can actually document that most of this activity isn't beneficial. It just kind of happens. And the way we know this is you can put proteins together with each other in a what's called a cell-free system. So you're not even doing this like in a cell culture. You're just in a test tube, right, in an environment where these proteins are stable, and you could just see like who sticks together when they bump into each other. And the answer is everybody sticks together, but most of it doesn't matter. It's not a stable interaction, so it's relatively short-lived, and you'll have a region of DNA where a protein grabs on, and then it lets go, and it grabs on again, it lets go again. It doesn't really do anything. It's just this transient biochemical interaction that happens. In Encode, they were trying to have the highest sensitivity they could, so they caught all that stuff, which is to their credit. That was the goal. Like, we want to be sensitive to this stuff. We want to know what's happening. But we have to recognize that a lot of these interactions are just proteins bumping into things and sticking for a second. The analogy I like to use in class for this, to get students kind of visualizing the inside of a cell, because you've probably seen like the figures in textbooks where it's like you've got the nucleus and the this, and it's like you've got like a few big things and then it's just like goop in between. That's not what a cell is like. The way I want everybody to think of the inside of a cell is, if you've ever seen like a swimming pool in like Brooklyn on 100-degree day, and it's just like bodies, just shoulder to shoulder, right? That's the inside of a cell. And all of those people are proteins and sugars and lipids, and they're just bumping into each other and interacting with each other to varying degrees. So you get a lot of these interactions biochemically, but most of them don't actually accomplish anything for that cell. They just happen because stuff is in proximity to each other. So that's a lot of what Encode is catching, are these very short-lived, kind of low-level activities that aren't contributing to the physiology of the cell. They just happen and then they go away.
But they might be doing something. Is that possible?
They might be. In a minute, we're going to talk about why I think they don't. So we'll get to, just to jump ahead just a second, we'll get to the 2014 Encode paper, and I'll talk just briefly about just a part of that figure. And it has to do with the interactions happen, but then why do we think they're not contributing biochemically? So we'll get there. We have to build up. There's a few things to build up to it. So the next kind of reason is, I mentioned sequence specificity, right? All of the proposed interactions we're talking about here require specific sequences, right? DNA binding to proteins to regulate expression, DNA or RNA interfering with a virus or with an mRNA to regulate gene expression, modification of the DNA for packaging. All of these things are specific DNA, protein, or like RNA interactions that are all sequence dependent, right? So if you have a region, again, that's not under purifying selection, we can say with confidence that it's not doing a sequence dependent function. That doesn't mean it's not doing a sequence independent function, but it's not doing something that requires a particular sequence, because if it was, there would be a selective pressure to keep doing its job. And we could directly document for most of the genome that there isn't. So the kind of takeaway from this one is, since over 90% of the genome is unconstrained, that 92-ish% is not doing any of the sequence-dependent jobs. It might be doing something else, but it's not doing the things where you need a specific sequence to interact in a specific way. And then we can look at mutations and degraded sequences. So this gets kind of towards how for each of those classes, I said there's a few that have been documented that like do important things. Well, this is why we can say for the rest, they're not doing those things. So we're going to use the endogenous retroviruses as an example of this. So there's about 200,000 sites in the human genome that are endogenous retroviruses. And endogenous retroviruses have a very specific signal that we can identify. They all have the same parts. All of them or some subset of the parts. So I mentioned they've got the duplications on either side, the target site duplications. And then in between those, this is what an ERV looks like. LTR stands for long terminal repeat. So you've got an LTR, and then GAG, POL, and ENV are the three genes for the virus, and then LTR on the other side. So those 5 components make up an intact, a complete endogenous retrovirus. And when the endogenous retroviruses have jobs, they need to use these components. So there are examples where an ERV sequence actually regulates the expression of a human gene. And it's in one of the LTRs that actually has the regulatory sequences that are responsible for that. And there's even at least one, maybe a couple, but there's one we can be really confident in, where we actually co-opted a gene from an ERV and we express it as part of our own gene expression profile now. So that was, I forget which one it is, but it was one of these genes that just straight up is now a human gene. Like we just straight up stole it from the virus and it's like, you're part of me now. So obviously those things are functional, right? But the vast majority of ERVs are so degraded, they can't do that stuff. So most of them are actually missing the genes entirely, and it's just either back-to-back LTRs or what's called a solo LTR, which is just the one LTR. In most of those cases, it's not the whole thing. You're missing part of that LTR, and it's also, like, mutated all the heck. So it's not doing the job. You can't co-opt a gene that's not there. And in order to do the regulatory function, you need to maintain the integrity of the sequence, and those sequences are degraded, they've experienced deletions and mutations. So the sequences for the vast majority of those 200,000 sites can't do the job that we've actually directly documented in some of them. So again, can I be certain that they're not doing anything? No. But can I be very confident that over 99% of those are not doing the function we've identified in some of them? Yes, they're not doing that function, the regulatory or gene expression function. Yeah. So again, certainty? No. But very high degree of confidence they're not doing any of the functions that have been proposed and sought out for ERVs.
Okay.
And then the last one here is expression levels. So a lot of the functions involve the RNA rather than the DNA in the genome itself. So for example, you are regulating gene expression by having like a pseudogene gets transcribed and that interacts with the mRNA for a protein coding gene, for example, and that either up or down regulates the rate at which you make that protein. Or you are transcribing an ERV and it interferes with a viral sequence, right? And it somehow interferes with that and it doesn't, the virus is unable to replicate in your cells due to that interaction. Now, a lot of the genome, depending on your source, it's 70 to 80%. I'm going to go with the 80% part of that range. About 80% of the genome is transcribed to the RNA at some point, at some stage in your life. Now, in order for that RNA to work, it needs to meet 2 conditions. It needs to have the right sequence to work. So right away, most of it, we know it's unconstrained. So right away, because only 8% of the genome is constrained, so right away, most of the RNA is going to fail that. But ignoring that, you need to have enough of that RNA to do the job. Because remember, we're talking about a packed swimming pool in the summer. So you need to have a bunch of things if you want to do a certain thing. You need to have enough of whatever chemical it is, enough of whatever molecule it is to do whatever the particular job is. And what Encode directly documents is that those expression levels, while covering a lot of the genome, the vast majority of them are extremely low. We're talking like 1 RNA molecule per cell or less, which is not a lot. That's not going to have any kind of impact on the physiology of that cell. It's just, there's just too much else going on, right? You're talking the amount of RNA in a cell at any given time, depending on your estimate, is going to be anywhere from the tens of thousands into the millions. So 1 single RNA molecule or 10 RNA molecules of a specific sequence in that pool is just too few to have an effect, right? So you need to have enough expression to matter. And when we look at the expression levels, they're mostly very low. So that's what I want to show you. But let me pause because I just talked a lot and see if you have any questions before I show the data.
No, I don't think I have any questions right now. I'm stuck on what was the term? Biochemically active?
Biochemically active versus functional. Yeah. Activity and
Yeah.
function are two different things. Yeah.
Yeah, that's a tough one for me. I'm gonna have to think about it some more because, yeah, it just biochemical activity to me seems super important.
So I'll give you an example of something that is biochemically active that I think we should have very high confidence is non-functional. You can have regions of the genome, and I'll say we can document this by tagging the RNA with fluorescence and then basically following the fluorescence in the cell.
Hmm.
So you can have a region of the DNA where you transcribe it. So you make RNA complementary to the DNA strand, and that's occurring in the nucleus of the cell because the DNA doesn't leave the nucleus. So you've got this little bit of RNA, and then before the RNA gets exported out of the nucleus, it goes through some quality control checks in your nucleus. And if it's not a competent RNA molecule, if it doesn't have all the parts, if it doesn't check all the boxes, then your cells are going to break it down and recycle it. You have enzymes in your nucleus that'll do that. So you can directly document that you can have an RNA transcript in your cell, and that RNA, the only thing it does is it immediately gets broken down back to RNA monomers and it gets recycled. And that's it. And that region of the genome, it won't get transcribed a lot, but every now and then you'll make a little piece of RNA and your cells will go, nope, not you. And it'll break it down and recycle those pieces. And so that's an example of biochemical activity, right? It's transcription, where it's not doing anything because physiologically for the cell, because the result of the transcription is immediately broken back down and recycled. So it doesn't even have the opportunity to like do any job for the cell. So that's an example of how you would draw a distinction between activity and function. And in the paper I'm going to talk about, this 2014 paper, they actually do a really nice job describing the conditions that would make you think it's one or the other, right? Activity with function or activity without function.
Okay, this is probably not a good example, but when you were describing that, my thought was, what if that's happening waiting for something where it would be needed, right? Like the thing that came to my mind was women and the cycle that they go through every month. If you didn't understand reproduction, you'd think there's no point in that. But there is a point once pregnancy happens. But if not, it's just going to be every single month, you know, you're going to have these, this egg and you're going to bleed. So anyway, that's what came to my mind.
Mm-hmm. So I think an understanding of the timing here might be relevant because the time spans that we're talking about would be seconds to minutes. So we're talking, you make the RNA and degrade it in the span of a couple of minutes is how fast this stuff goes. So you wouldn't have the opportunity to basically like have the RNA and then, oh, hey, the thing that it interacts with is here, let's use it. Like you wouldn't even have that opportunity because it's there and it's gone, just like immediately. And the way you would regulate if there's something, so like what you kind of described is like a gene regulatory mechanism, but typically the way this works is that molecule appears in the cell, and that molecule will interact with something, and that will lead to a change in the genome. So like a particular sugar, for example, is present, and then that leads to the expression of the enzymes to break down that particular sugar. Right? You're not expressing those enzymes all the time and degrading them. You're waiting for the target to be present, and then once it is present, now you can make that gene product. Or the other thing our cells do is we'll make the proteins, we'll make the enzymes, but we'll keep them in an inactive state until we need them. And then you could have a signaling cascade that rapidly activates the proteins when we need it so we could be responsive to a change in conditions. You wouldn't do that, but you wouldn't do that with the RNA. You would do that with the polypeptide at the other ends of the process. The only situation I could think of where you actually do something like that with RNA is in the egg cell, because at fertilization, you activate a bunch of gene expression of RNA that's already there. So they did transcription, now you flip the switch and you do translation really fast so that you have all the proteins you need to kickstart embryonic development. But that's not rapidly transcribe, break down, transcribe, break down. It's you transcribe that stuff before meiosis, and then it just sits throughout the process because you have no opportunity for transcription during. So it's not a perfect one-to-one there, but that's the only thing I can think of that's kind of close to what you're describing. Yeah.
Okay, that's helpful. Thank you.
Okay, so let's look at the Encode data from, this is the 2014 Encode paper. The key thing here is, I know this is like a lot going on in this figure, so what I'm going to say is just like, don't worry about that stuff, because that stuff is protein binding, proteins associating with the DNA, and don't worry about it. We want to talk about RNA and transcription, because again, like 80% of the genome-ish, not quite, in this one it's 76% or so is where that lines up, is transcribed at some level. But the colors here indicate the level of transcription. So the darker the color, the more it's transcribed at very high levels. And if you go through and dig through the methods of this paper, which I have done, and dig through the supplemental data of this paper, which I have done, and it took a really long time to track those figures down, what you can see is that this is a weird measure. I don't even remember what it stands for now, because I did this a couple years ago. It was FPKM. It doesn't matter what it stands for. Basically, it's how much of the RNA is present in the cell. And these bar graphs broken up by category tell you for each region of the genome how much of that area that's transcribed is present at a certain threshold. So this stuff down here at like the 500, 150, 10, that stuff is definitely biochemically relevant to the cell. Like, we really care about that stuff. By the way, you could ignore these two columns, because this column, this third one, that's all RNA. That's the one we care about. Just lump it all together and just get the maximum, extent that you can find. And what we care about here, if you read the figure legend, somewhere from 5 to 1 indicates one copy of the RNA molecule per cell at any given time. So where they detected the RNA, there was at least one copy. But if it's in that like 0.5, 0.1, greater than 0, but not 0.1, that all indicates that the transcription level is less than one RNA molecule per cell. And I had a months-long conversation on this with Dr. Casey Luskin from Discovery Institute, and one thing he brought up was these are averages. So you could have a gene that's expressed in a specific cell, or I should say a gene, a region that is transcribed in a specific cell, or that is only transcriptionally active in a region in a specific time during the life, and you see this big spike at that time that's enough RNA to be functional, and then it's like 0 the rest of the time, so that's going to average out to very low, even though in the assays where it was detected, it was actually very high. That is a reasonable objection to make to what I am saying here. So in response to that, I dug through pages and pages of the supplemental data for this experiment, looking at the individual assays. And actually, now I forget the numbers, but it was less than 1% was present at more than one RNA transcript per cell for the individual assays. So it wasn't the case where you had like super high levels in one and then 0 for 1,000, and therefore it was, very low average. It was like the vast majority were very low. So I feel like I'm in the clear in my characterization of these data, because I really dug into it way back when. So that's Encode. Now, in this same paper, I will say, it's a long paragraph, so I didn't put it in the slides, but the authors of this paper actually do a really nice job describing basically what you would expect to see if a region is functional and if a region is not. And the specific criteria they gave to say, if you see these things, this region is probably not functional. They say low levels of transcription, low levels of constraint. And the vast majority of the human genome checks those two boxes, which is another avenue to being able to confidently say most of the genome is not functional. And that's not me saying that, that is Encode saying that in their 2014 paper. So that is the last thing I wanted to show.
Okay.
The only thing I have left is the summary.
All right. I have a couple things.
Go for it.
Why? I'm guessing you're going to say there's ethical concerns, but why don't we just test this?
Yeah, you can do like deletion experiments in mice, but the ethical, you can't do deletion experiments in people.
What if someone volunteers?
You probably still couldn't, because you can't volunteer to do something that there's like a reasonable concern that it might hurt you. And deleting stuff from your DNA is a pretty good way to experience some problem. We can do it in cell cultures, but we can't do it in like adult people.
Okay. Now what you just said seems to contradict what you just taught me for 2 hours.
Yes, because we have a really good understanding of what's in the genome and what goes where and how it interacts, but we do not have a complete understanding of what's in the genome and what goes where and how it interacts and why it matters. So there is almost a 100% chance that if you were to identify, say, a region in your genome that's, like in the mice, it was 30 million base pairs, there is close to a 100% chance that if you were to identify a candidate region in the human genome where you have 30 million nucleotides and say, we don't think there's anything in there that's doing anything. It's this region of repeats, and it's this region, it's this. There's very likely something in there that's still interacting in some way that matters, right? Because these interactions are scattered throughout the genome, and it may literally be 10 nucleotides. But those 10 nucleotides matter. And if you get rid of them, that causes, some problem for chromatin structure and whatnot. The likelihood of it like killing you, quite low, especially because it obviously wouldn't be in all your cells, right? This would be like this would have to be some weird kind of gene therapy and only be in some tissues or something, right? But I think I just saw a comment on here, and it went by, oh, here it is. Yeah, from Azaria. Here's the comment. There's no way to have informed consent, right? If you volunteer for a drug trial, you can have informed consent, right? Here's what the chemical is, here's how it interacts, here's what we expect, here's what we saw in the phase 1 trials, whatever, right? You can have informed consent there. We're going to delete X amount of your genome. We did the same deletion in mice. Here's what happened in the mice. It was fine. I don't think that would pass a review board. Our standards for experimenting on humans are extraordinarily high, rightfully so.
Wouldn't it make sense then to do it on chimps?
Yeah, we could do it on chimps probably.
Okay. That would be cool.
I bet somebody is.
All right.
Yeah.
I have a friend, and her son has a genetic disorder that's very serious. And the doctors are telling her that it is from, it's essentially a mutation in the non-coding region. And so am I hearing right that they're telling her that this genome that her son doesn't need is what caused this disorder?
Not necessarily. It depends on the condition. So a non-coding region could be an enhancer, a silencer. It could be a promoter. It could be a scaffolding protein attachment. It could be a methylation. I mean, there's a million things that it could be that are important that are non-protein coding.
Does evolution state though that in the non-coding regions that you're claiming are non-functional, you can get a mutation that can cause a disease or disorder?
Yes, absolutely. So one, yes, but also you could have a mutation in a non-coding region that's still functional in some way. You can also have a mutation in a non-coding, non-functional region that causes a biochemical activity to occur that shouldn't be occurring. And I'll give you two examples, one hypothetical and one real. So the hypothetical one is you have one of those mobile genetic elements, a transposon, a retrovirus, whatever, and you are methylating it, and that's keeping it immobile in your DNA. And we do this for those viral sequences. If it mutates such that it loses the methylation site, now it could potentially mobilize because it's now open to being interacting with enzymes. And then that mobilized sequence could cause, it could insert itself in a gene that then breaks. Or if it's a virus, now you've got a virus that's doing its thing, virusing all in your cells, and that's a problem, right? So you could have a region that's non-functional that becomes biochemically active due to a mutation. Another example of this is Huntington's disease. That's a region where it's non-protein coding, it's actually just repeats. The length matters. So if you have, I'm gonna forget the numbers. I think it's like under 40 of the repeats, then you're fine. If it's 40 to 60, then you'll, maybe experience some symptoms later in life. If it's above 60, you're going to get Huntington's disease. And that region is non-protein coding. It's not like an enhancer or a silencer. It doesn't code for an RNA that does. It's just the length of that region matters. And if it's too long, then you're out of luck, right? So there's lots of examples where you can have problems in a non-protein coding region, whether that region is functional or not, that have adverse downstream consequences.
Okay. Okay. Thank you.
You're welcome. All right. So this is the sorry. This is the kind of takeaway from this Kellis paper that I described, that low transcription plus low conservation, probably non-functional. That's what the authors kind of describe. And in their own figures, that describes most of the genome. So we can summarize this. 10 to 15% of the genome, definitely functional. I think more accurately you would say like 8 to 12, but for today we'll say 10 to 15%. Mm-hmm. About 80% of the genome cannot do any of those documented functions because it's unconstrained, it's got deletions where it's like missing functional bits that it would need, right, to do those things. So we have 10 to 15% of the genome, we can say, here's what it is, here's what it does. That's functional. About 80% of the genome, we can look at it and go, it can't do any of those things that we just documented in the 10 or 15%. It lacks purifying selection. The sequence is either too mutated, it deviates too much from what it would need to be functional, or it's incomplete, it's experienced deletions, it's missing parts, or it's transcribed at levels that are too low to matter physiologically. Right? And 80% of the genome meets these criteria in some way, right? About 90% of the genome meets this. This is going to be 75% of the genome is in that boat right there. And then the two mutated or incomplete is virtually all of the ERVs and transposons. So most of the genome meets those criteria, which means that it could still be functional. I want to be crystal clear about this. That is not to say that that 80% is not functional 100%, take it to the bank, right? It's saying that that 80%, we can very confidently say it's not doing any of the functions we have identified thus far in our study of the human genome, and it is not doing any of the functions that have been proposed for junk DNA due to the fact that it lacks selection, it's degraded and missing pieces, and if it's transcribed, it's at very low levels, right? That, again, doesn't mean it's non-functional. It means it's not doing any of the functions we've identified.
Okay.
And that's it.
All right. All right, cool.
We made it.
Yes, let me end with this.
Yeah.
I think that if you guys are right about this, this is a very compelling argument. I think this would be enough to change my mind. But this is something I learned with my Flat Earth, the final experiment, is that predictions that end up not happening make it harder to convert people. And so evolution does have a history of bad predictions. And so I do want to watch this one carefully. But I do think that this is compelling if true. I also just shared my screen for something real quick. It's pretty cool, even though I don't understand it.
Here we go. Okay, what are we looking at?
This is brand new. This came out 6 days ago.
I haven't read this, but I've read about this. Yeah.
So this came out 6 days ago. Fortunately, someone sent it to me. And something that they believed was non-functional, they just found a purpose for it. So these things are going to potentially come out in the future. And so as the saying goes, time will tell.
Yeah, so there's a bunch of stuff about this recently, looking at the correlation between genome and cell size, and a bunch of different things there. And so I haven't read this one, so I can't comment specifically on this.
Mhm.
But in general, the stuff with genome and cell size is, I think, based on what we've documented, putting the cart before the horse. Because what we have directly documented experimentally is that you can't select against a lot of this DNA that builds up in your genome. So rather than the DNA being like functional and affecting cell size, is that the cell size is a response to the fact that we have DNA that's just kind of there and we have to deal with it.
Mhm.
It's akin to not having any garbage collection, so you have to like have a storage bin where you put your garbage, right? You have to take an extra storage, so, like, you just have to have more space for all your stuff because you can never get rid of it, right?
Mhm.
If you could never have a yard sale, you're eventually going to need to get a storage unit somewhere for all your stuff because it'll start exploding out of your basement windows, right? I think that's what's going on here. That the cells are responsive to the amount of DNA. That's not a function of the DNA. That's akin to the use an analogy someone brought up before, the gum on your shoe, and the cells
Mhm.
are kind of having to deal with the fact that we have all this DNA and we can't get rid of it. I think
Mhm.
that's what's going on here. But I
Okay.
haven't seen this one specifically, so I can't comment specifically on that one. But there have been a few years worth of that kind of stuff floating around with cell size and junk DNA.
Okay, cool. People in the comments are saying they don't know about evolution's failed predictions.
So I actually made a note to myself that, like, if you want to chat again, we could do a whole show on evolutionary predictions, because let me tell you, there's some really cool ones in there. There's some really cool evolutionary predictions.
Yeah.
And there's yeah, it's there. The list of confirmed predictions is really awesome. And there's some really cool stuff of just like, here is a thing that we couldn't check at the time because we didn't have the technology. And then one example of this, and this is not to go into a whole thing, but just one example of this is in the 1800s with the famous peppered moth thing. There was a population geneticist that analyzed the original data. So the experiment was like in the 1800s, and then in the early 1900s, like once like population genetics existed as a field. I forget exactly when they did this math, but they calculated some parameters about how that trait would work. And everyone looked at that math and went like, there's no way. That's totally wrong. Like, there's no way that's realistic. Your numbers are wrong. You're completely wrong. In like the 19 Zach Hancock is going to get so mad at me if I get this wrong, but I think it was in like the 1980s. They went and redid the experiment, but with modern technology, so you could actually sequence the genomes. You could do more precise stuff once you're doing it in the late 20th century as opposed to the 1800s, early 1900s, and the numbers were spot on. All those numbers from the 1920s that they were like, there's no way, that's so wrong. They were dead on. And
Wow.
it's just this beautiful example of where we didn't even have the ability to check our work at the time. We were just doing math and saying, this must be what's happening.
Hmm.
And then half a century later, we finally have the ability to like check our numbers, and it was bang on. So it was,
Wow.
there's some really cool stuff out there like that.
I'm being called out in the comments to name what I'm talking about. I can do it very briefly.
Yeah, go for it if you want.
And again, people need to understand what I'm trying to say right now. All I'm trying to say is that it makes converting someone, it raises the bar. That's all I'm saying. So please hear me out. I'm actually helping you understand how to convert someone who doesn't agree with you. You just have to do a little bit more work. So number one, ontogeny recapitulates phylogeny. That was wrong. And that was something that was predicted based on evolution.
Mm-hmm. And
Number
we
two,
kind of gave that one up pretty quickly, though. I know
okay,
textbooks were behind, but we gave that one up pretty fast.
okay. Number two is vestigial organs. Growing up, I can't tell you how many classmates came in and were like, I'm getting my tonsils out. And I'm just like, what? And I'm like, I don't know if I don't even know what my tonsils are. And so the prediction that we're going to have organs that are left over from previous whatever that we don't need anymore, that was wrong. And that actually affected millions of children. That one affects me personally.
So that actually relies on a kind of a there's different it's like we talk about the definition of functional. There's also different definitions of vestigial.
Hmm.
Vestigial, if you're being specific about it, vestigial doesn't mean it has no function. It means it's lost an ancestral function, right? So
Hmm.
the appendix, for example,
Hmm.
still
I was
has
going
a
to bring that up
role.
too.
Yeah, it still has a role in immune functions and regulating the gut microbiome, but it used to be a digestive cavity, which it no longer is, right? So it has lost an ancestral function. Still has other jobs it does, but has lost the ancestral function. Another one, take a look at your wrist right here. This was my favorite they do in class. Everyone, you can take a look at your wrist. See this? Sorry, this hand right there. See that tendon right there? That is vestigial. We don't use that for anything. Most people have two. Some people only have one. Rarely somebody has none. That is the tendon that is apparently it's an ancestral thing in whatever clade we're in mammals if you go back far enough, and it's for retractable claws. But we don't have retractable claws. We have wimpy fingernails instead. But we still have the tendon for them. So that's a I like that one.
Yeah, on the appendix, people would have like doctors would be doing a surgery,
And they just grab it. Yeah,
and they're like,
yeah,
"Hey,
it's dumb.
while they're in there, we're going to take it out because it's vestigial."
Yeah, it's
So,
dumb.
okay, the third one is bad design. Um,
Yeah, I
the
don't
the
like that
one
one, but
the one that I heard the most often was that the human eye was a bad design, and that ended up being false.
well, it does. It's true that it has a blind spot, which other eyes don't have, which is not great. Because you could have the optic nerve behind instead of in front. So it's true that we do have a blind spot, and other versions of eyes don't have it. But my go-to, I don't like the bad design argument, but it's
Mhm.
the way I think about it is evolutionary constraints. So like ankles are a good example of this because ankles are wrists that were weight bearing. If you've ever done like a handstand for more than 10 seconds, you know that this structure is not good at bearing weight. And in particular, it's very unstable laterally. But we walk on two of these, and they're that's why it's really easy to roll your ankle. And like so that's I wouldn't call that bad design. I would describe that as like that is a an evolutionary constraint. Right, you don't start with a blank slate. You're constrained by your history, and you end up with things that are suboptimal for the role that they play. Right,
Yeah, I
but
don't remember what was discovered, but it's like molar glial cells.
in the eye,
Uh, yeah, whatever was stated as an argument against creation about the eye ended up being wrong. So
okay.
those are those are three things that I would say were wrong. And again, it doesn't mean evolution's wrong. It just means when there's a pattern, someone who disagrees is naturally going to remember those things.
I will say that those are I don't know that I would call those necessarily predictions,
Hmm.
right?
That's fine. Yeah.
That's vestigiality is you could observe that it doesn't have if you're hypothesizing common ancestry, then you observe something doesn't have a function that it did in the past, then you assign the label vestigiality. That's not
Hmm.
to say it doesn't have any function; it just doesn't
Hmm.
have that job, right? The Haeckel embryo ontogeny recapits phylogeny thing. It's not true. It's what it really is that it's you develop the ancestral traits first and the more derived traits for your clade later in development. So earlier on, organisms across broader groups look more similar because they're developing like the vertebrate traits. And then the mammal versus the reptile versus this versus that, those specific traits come later and they start to deviate from each other, right? It's not that you have a fish stage and a frog stage and a lizard stage and this. That was dumb.
Hmm.
But that also like was it was stupid that it was in textbooks. It shouldn't have been. But it was never. That was more PR than like good science. And trust me, you should hear me talk about scientists I don't like. I'm the first person to point out shoddy work by other scientists.
Wow.
We you look. There's another evolutionary biologist on YouTube, Dr. Jay Bundy, was once challenged. Well, so and so says this, and he said, "I am an evolutionary biologist. It's my job to argue with evolutionary biologists. Like, I'm not afraid to call these people wrong. That's yeah, that's oh yeah. Academia is vicious. Don't no, knives will be out. Don't worry."
Yeah. Cool.
All right, let me we did get a bunch of super chats. Let me just see if there's any like actual questions in here. So we got Roshana 499. Junk DNA is a strict subset of non-coding DNA. I would say it's a strict. I can't even say that because we have documented very small number, but there are a number of junk proteins where it gets, like I described with the mRNA or with the RNA, and it gets broken down right away. There are polypeptides that do that. So I can't even say junk is a strict subset of non-protein coding. There's a very small, it's bio. There's exceptions to everything. It's going to be true 99% of the time, but not 100%. Yeah. Okay, orthogenes. This is back to vitamin C. Yeah, there we go. Okay. It's a recommendation to check out Stated Clearly on herbs. Yeah, the herb stuff is a fantastic bit on human chimp common ancestry specifically because of the shared herbs in the same places. And this is the really cool part, mutations that are the same. So you could have like a human, chimp, and gorilla have the same herb in the same place, but then the human and chimp share a mutation that the gorilla doesn't have. So it shows you the pattern really neatly. So yeah, definitely recommend checking out Stated Clearly on that. He has some good stuff on that one. Let's see. Let's see. We got Doug Meeks. God could have designed it. Process of evolution and through providence. And
Yeah, I agree with that, actually. I definitely agree with that. I can't reconcile no God at all. That makes zero sense to me, but I could absolutely reconcile God with evolution, which is why it's such a popular view.
yeah, I know a lot of people that are the vast majority of people who accept evolution also believe in some being, however, whatever you want to label it, vast majority. And then doing knockout tests. Oh, this is a good point, actually. Knockout tests on humans, because we have long generations, so it's actually not super helpful. That's a good point. Yeah, that's a useful point. Is that mice you can do several generations a year. Humans, you're gonna wait a long time to see if the grandkids are okay. Yeah, that's a good point. That was everything.
Cool.
All right, I said at the front end this was going to be a short show, and here we are 2.5 hours later. Sorry to keep you this long, Will.
It's okay. It was great. It was great. You gave me a lot to think about. I will watch it again and digest all of this. And again, I'm going to admit that this is a compelling argument if true.
Okay, well, I appreciate you being here, and I'm looking forward to. I think it's January 7th. You got the next installment with Erica. I'm
Yep.
looking forward to that. And then, folks, just a quick update what we've got going on here. I know you've been waiting with bated breath. On Wednesday, the 24th, I'm finally, at long last, releasing that video on Winston Ewert's dependency graph model. It is almost an hour long, so that's why it took so long to get it out, because it took a long time. So that's what I've got, and that takes us almost through the end of the year. So thanks for being here, everybody. I appreciate you all. Thank you. I think everybody was mostly behaving in the chat. I didn't see too many issues. So thanks, everybody. And I will see you next time I do a live thing. I don't know when that's going to be. Bye, everybody.